Hello and welcome to Chapter 8. In this chapter, we're going to talk about microbial metabolism, and we're actually going to talk about one of the processes of metabolism that actually produces this stuff right here. If anybody knows what this is, think about it. This is what's known as kimchi, and it is a fermentation product with cabbage, and we'll talk about the process of fermentation. So, what is metabolism? Well, metabolism is the sum total of all the chemical reactions and workings inside of a cell. And we can divide metabolism into two major groups. Anabolism, which is biosynthesis. It is the synthesis of cell molecules and structures. Anabolism requires the expenditure of energy. Catabolism is the exact opposite. It is the breakdown of bonds of larger molecules to actually release energy from those bonds. Metabolism accomplishes the following things. One, we can assemble smaller molecules into larger macromolecules for the cell, utilizing ATP as the energy source. Also, metabolism will degrade macromolecules into smaller molecules, a process which will break chemical bonds and yield energy through the process of catabolism. And metabolism will conserve energy in the form of ATP or heat. Let me make this statement. The second law of thermodynamics states that energy can neither be created nor destroyed. It can only change forms. So when I say something about we're going to make ATP, actually what I'm saying is that we're going to convert one form of energy into ATP within the cell through the processes of metabolism. This chart just simply shows that, for example, glucose is one of our main nutrients from outside the cell or from internal pathways within the cell. And this large molecule that has 6 carbons, 12 hydrogens, and 6 oxygens have to be broken down by catabolism. And we do that through the several processes of glycolysis, the Krebs cycle or the citric acid cycle, the electron transport chain, or fermentation. All of these will break bonds, and that will yield energy. That energy in the form of ATP then can actually be used to take precursor molecules such as pyruvic acid, acetyl-CoA, or glyceraldehyde 3-phosphate, also known as G3P, through the processes of anabolism and make larger molecules. So we can take amino acids, the simple sugars, the nucleotides, and fatty acids, and again using more energy, build proteins, the peptidoglycan that bacteria will need, the nucleotides with the RNA and DNA, and fatty acids along with some sugars to make the complex lipids. And all of those macromolecules together, along again with some energy for assembly, will make up the bacterial cell. Now, what are enzymes? Well, enzymes actually are catalysts. They speed up the rate of a chemical reaction without becoming part of the products or are consumed themselves in the reaction. Enzymes come out of the chemical reaction unchanged. Enzymes are biological catalysts that overcome the activation energy needed for a chemical reaction to occur and allows that chemical reaction to proceed by either increasing the thermal energy or heating to increase the velocity of molecules or to increase the concentration of the reactant molecules to increase the rate of molecular collisions. The collisions actually then allow for the chemical reactions to occur. Enzymes are mostly composed of proteins. In fact, I will just make the statement that all enzymes are proteins or at least originate from proteins. Now, are all proteins enzymes? No. Most enzymes do require some sort of cofactor or coenzyme, and we'll discuss that here in a few moments. These enzymes act as organic catalysts to speed up the rate of cellular reactions by lowering the activation energy required for that chemical reaction to proceed. They have unique characteristics such as shape, specificity, and function. They enable metabolic reactions to proceed at speeds compatible with life. They have an active site for the target molecules known as substrates. They are typically much larger in size than their substrates, and they are usually associated closely with substrates but do not become integrated in the reaction products. So again, they come out unchanged by the chemical reaction. They can be recycled, thus function in extremely low concentrations. However, they are greatly affected by temperature and pH because they are proteins, and they are typically regulated either by positive or negative feedback mechanisms or by genetic mechanisms. So how do enzymes work? Well, again, the substrate is the reaction molecule upon which the enzyme acts, and typically every enzyme has its own unique substrate. Enzymes bind to the substrates and participate directly in changes to that substrate, but again, they come out unchanged at the end of the reaction. So they do not become a part of the products of the chemical reaction and therefore are not used up by the reaction. So they can function over and over again as long as there is substrate present in the environment. The protein molecules that make up enzymes are actually known as the simple enzymes, and that's the protein structure alone. These are also sometimes known as apoenzymes. Conjugated enzymes, or what's known as holoenzymes, contain not only the protein structure, or the apoenzyme, but some other non-protein molecule. And those are either cofactors, which are non-protein portions, or coenzymes, which are some sort of small organic cofactor. So the difference between a cofactor and coenzyme is the cofactor is inorganic and the coenzyme is organic. So this picture just simply shows, here's our apoenzyme, and it has its metallic cofactor attached. So this would now have an active site somewhere, or it can have this coenzyme attached to the apoenzyme, or could have both a cofactor and coenzyme that are necessary for the activation of the enzyme to its holoenzyme and active structure. So again, apoenzyme simply made up of proteins. So we go from a primary amino acid chain to secondary structure to tertiary structures, and then finally usually to quaternary functions. Most complex enzymes have a quaternary structure consisting of several polypeptides bound by weak forces, and often the active site is formed by the junction of two of these polypeptides. So here would be an active site. The holoenzyme is the active enzyme. So the active site is able to bind to its substrate. Usually these active sites are three-dimensional crevices or grooves formed by the way the amino acid chains fold within the protein structure. So each enzyme has a different primary structure, the amino acid sequence, variation in folding that is due to the primary structure, and this gives us unique active sites for each enzyme. So here's our holoenzyme ready to go, and these are two substrates. And you'll notice that this substrate will fit nicely into the folding of the protein, while this one does not fit at all. So the substrate for this enzyme will fit into the active site, forming what's known as the enzyme substrate complex. This will strain bonds within the substrate, causing the reaction to occur, and we get our products, but again, the enzyme comes out unchanged at the end of the reaction. Cofactors typically are metallic, and so oftentimes they're metallic ions such as iron, copper, magnesium, manganese, zinc, cobalt, calcium, etc. They assist in the precise function between the enzyme and the substrate. The cofactors are necessary to bind with the apoenzyme in order to activate the active site. So these cofactors will help bring the active site and the substrate close together for binding. and oftentimes these cofactors will participate directly in the chemical reactions. Coenzymes are small organic compounds that work with the apoenzyme to alter the substrate as well. So coenzymes are also necessary to activate the apoenzyme to its holoenzyme form. Oftentimes these organic compounds will help remove a chemical group from one substrate and add it to another substrate. They can carry or transfer hydrogen atoms, electrons, carbon dioxide, and amino groups between different substrates. Many of our vitamins are actually very important components of these coenzymes. There are functionally 6 classes of enzymes. The oxyreductases. These actually transfer electrons from one substrate to another, and dehydrogenases transfer hydrogen from one compound to another. So a dehydrogenase is actually a form of an oxyreductase. Transferases transfer a functional group from one substrate to another, and hydrolases cleave bonds or break bonds on molecules by adding water in order to break the bonds. Lyases add groups or remove groups from double bonded substrates. Isomerases actually are going to convert a substrate to its isomeric form or its mirror image form, while ligases catalyze the formation of bonds with the input of ATP and the removal of water in order to form bonds between atoms. And you'll notice all of these end in ASE. ASE, or the suffix ZYME, means that these act as enzymes. So here's just a sampling of enzymes, their substrates, and their reaction. Lactase, for example, its substrate is lactose, and the function of lactase is to break lactose down into its two smaller, simpler sugars of glucose and galactose. Penicillinase, we'll talk about later on, also is known as beta-lactamase, actually will break down the beta-lactam ring in the antibiotic penicillin, making it therefore ineffective. DNA polymerase actually is going to work on DNA nucleotides, and basically DNA polymerase synthesizes DNA. Exoenzymes are made in a cell, but then are transported extracellularly where they have their effects. So oftentimes these are digestive enzymes that break down large food molecules or harmful chemicals outside of the cell. Endoenzymes are retained intracellularly and function there. Most of the time, endoenzymes are actually enzymes of metabolic pathways. This picture just shows, again, the inactive apoenzyme is made inside the cell and transported outside, where it then usually is activated by its cofactor or coenzyme and then combined to its substrate to release the products extracellularly. Whereas endoenzymes stay inside the cell and act upon their substrate within the cell. So how do we regulate enzyme function? Well, there's basically two forms of enzymes. The constitutive enzymes, which are always present in relative constant amounts within cells, regardless of the cellular environment. Whereas regulated enzymes are usually turned on or induced, or turned off or repressed in response to changes in the concentration of the substrate within the cell. So here's the constitutive enzyme. Constitutive enzyme, as you'll see, there's 5 of them represented in this cell. And so how many of the substrates can we deal with at any given time? Well, we can only deal with 5. So does increasing the amount of substrate increase the amount of enzymes in constitutive enzymes? The answer is no, and we can still only deal with 5 substrates at a given time. However, regulated enzymes, if we add more substrate, we actually wind up making more enzyme so that we can operate on a lot more substrate in any given time. Or if the substrate actually declines in concentration, then many of these enzymes will be broken down and recycled so that we only have a small number that's present at any given time. Transfer reactions by enzymes are accomplished by oxireductases. Oxidation is the loss of electrons from a molecule. So a compound that loses an electron is oxidized. Reduction is the gain of an electron or electrons. So a compound that gains electrons is reduced. And there is actually a mnemonic, oil rig. Oxidation is losing, reduction is gaining. NAD+, NADP+, and FAD+ are coenzyme carriers of electrons, and we'll talk about these more in just a little bit. Pathogens secrete unique exoenzymes that help them avoid host defenses or promote multiplication in tissues, and this is what can make them so deadly sometimes. They are considered then virulence factors or toxins because they contribute to the overall disease process. Examples of these are streptokinase, made by the Streptococcus bacteria, streptolysin, elastase, collagenase, lipase, and penicillinase. Now, since enzymes are primarily made from proteins, they can easily be denatured. The weak bonds that maintain the shape of an enzyme in the protein component are oftentimes broken down easily by heat, either low or high, so you can have too much heat or too cold, pH, or certain chemicals. And this denaturing disrupts and distorts the enzyme's shape so that it can no longer bind to its substrate. Non-functional enzymes block metabolic reactions and can actually lead to cellular death. So let's talk about the metabolic pathways. Metabolic reactions most often occur in a multi-series or pathway, or sometimes referred to as a cascade or cycle. Each step will be catalyzed by an enzyme, but we're not going to go through each enzyme associated with every step through these metabolic processes. If you want to learn all those, go take graduate biochemistry. The product of one reaction is often the reactant or substrate for the next enzyme in the series. Many pathways have branches that have alternative methods for nutrient processing. Some pathways will take a cyclic form, and these pathways are usually interconnected and merge at many different sites along the way. So we can look at multi-enzyme systems, and a good example here is a linear pathway, so that reactant A is converted to product B, which becomes reactant B, that's converted to C, to D, to E, etc. And a good example of this is actually glycolysis that we'll talk about here in a few moments. Cyclic multi-enzyme systems do exactly what the name says. They cycle. So when U enters and combines with V and T comes in, we make W, X, Y, Z, but Z then will actually go back to V. So we have this cyclic nature of the system. And the Krebs cycle or citric acid cycle is one of these examples. And then we can have branched multi-enzyme systems, and they can be either divergent, where M goes to N, and then we have a choice. And whichever enzyme is present in either the greater amount or just simply present there, then that may determine, does N go to O and down this way, or does N go to P, Q, etc. And protein catabolism is a good example of a divergent branch multi-enzyme system. Or the system could be convergent, meaning that A, B goes to B, goes to C, X goes to Y, goes to Z, and then C and Z actually are necessary to make M, which then becomes a common pathway. A good example of this actually is the hemopoiesis the hemopoietic system, our blood clotting system. So we have one line that goes this way and one that comes this way. We come down to a common denominator and then we go down from there. But another good example is the amino acid synthesis in building a protein. Now, direct control of the action of enzymes is done through inhibition, and this can be either competitive or non-competitive. Competitive inhibition is where a molecule that resembles the substrate occupies the active site of the holoenzyme, preventing the substrate from binding. The enzyme cannot act on the inhibitor and is therefore effectively shut down. Non-competitive inhibition, some enzymes have two binding sites, a binding site and a regulatory site. The regulatory site can be regulated by binding of molecules other than the substrate to the regulatory site. What this actually does then is actually change the shape of the active site so that the substrate can no longer bind. So oftentimes the regulatory molecule is the product of the enzymatic reaction itself, and this provides a negative feedback loop that slows enzymatic activity once a certain concentration of the product has been reached. So here are two things competing for the active site in this enzyme. One is the normal substrate, and if it gets to bind to the active site, then the chemical reaction occurs. But if the competitor binds to that site, then basically the enzyme is shut down at that point. Non-competitive inhibition, so here's our active site, and again, as long as the active site and the substrate fit together, we'll get the product. But what if this product is the regulatory molecule? It then will bind to the regulatory site and change the shape of the active site, causing the substrate not to be able to bond, and the reaction therefore is blocked. Enzyme repression can occur, and this will stop further synthesis of the enzyme somewhere along its pathway. If the end product of the enzymatic reaction reaches excess, the genetic apparatus for actually replacing the enzymes is going to be repressed. The response time is longer, but the effects of repression are actually more enduring through this process. Enzymes appear or are induced in regulatory enzymes only when the suitable substrates are present. It takes energy to make these proteins, so the cell is only going to make the protein enzyme when it's necessary. So synthesis of the enzyme is induced by the substrate being present. It's really an inverse of enzyme repression. So, where is the instructions for making proteins? Well, that's found in the DNA, and we're going to make a copy of the gene for that enzyme to messenger RNA. That will then be translated into the protein, into the amino acid chain, and the structure of the chain will cause the enzyme to fold up into its functional origin. That then can bind to the substrate, and that will produce our products. But if the product becomes in excess, it can actually bind to the gene on the DNA, effectively stopping then translation of that gene to messenger RNA. And so if we don't get messenger RNA, we don't build the enzyme protein, no enzyme, and so we don't actually expend energy in making this protein if the substrate is not present. So concept check. A cell uses what to obtain energy and what to build macromolecules. A holoenzyme is composed of what two functional portions? And if an enzyme is rendered inactive by change of temperature, change in pH, or chemicals, it is said to have been what? And in what type of enzyme inhibition does a regulator molecule bind to the regulatory site and change the shape of the active site? So while we have catabolic and anabolic reactions in the cell, they can both be one or the other of these types of reactions. Exergonic reactions are those reactions where energy is going to be released as the reaction occurs, and this is normally due to the breakage of bonds between atoms. Energy that is derived from these exergonic reactions then are available for doing cellular work. While endergonic reactions are the exact opposite. These require the addition of energy to move forward, and usually this is the formation of bonds. Exergonic and endergonic reactions are usually coupled. And so, for example, when we break down glucose and we have a high amount of energy in all of these bonds as the electrons are moving within their orbital shells. And as we break these bonds through catabolism, through anaerobic reaction, we are going to get a huge amount of energy that we can then convert to ATP, and that ATP then is used to perform cellular work. Redox reactions always occur in pair, and these are known as redox pairs. You always will have an electron donor and an electron acceptor. Oxireductases are important in the cellular redox reactions, and the coenzyme carriers of NAD+, NADP+, which is found in photosynthesis, and FAD+, are going to be very important in carrying the hydrogen ions and electrons. So a reducing agent will give up the electron, and so sodium, when it loses the electron to chloride, is actually going to be oxidized, because oxidized, remember, is losing the electron. Chloride accepts the electron that sodium gave up, and therefore it is going to be reduced because it gained the electron. So when NAD plus gains a hydrogen and the electron from the hydrogen ion that'll be formed here to form NADH plus the hydrogen ion, what we're going to see is that the oxidized NAD plus will then go to the reduced form of NADH, and then this hydrogen ion will go along with the NADH. However, we usually don't write the hydrogen ion because we know that it's there, and so we just write NADH. Phosphorylation is the energy captured in the electron carrier is used to then phosphorylate or add inorganic phosphate to adenosine diphosphate or another compound. And when we add a third phosphate to ADP, we get ATP. This will then store the energy in the high energy bonds between phosphates in ATP. NAD plus is the most common electron carrier that we see. It carries hydrogens and electrons from dehydrogenation reactions, and the reduced NAD plus will be NADH plus the hydrogen ion, but again, we rarely actually write the hydrogen ion. We simply remember that it's going along with the NADH. Catabolic pathways are where we see electrons being extracted and carried through a series of redox reactions to a final electron acceptor. And this is going to be primarily the electron transport chain. In aerobic metabolism, oxygen is the final electron acceptor at the end of the electron transport chain. In anaerobic metabolism, an organic or inorganic compound, not molecular oxygen, will be the final electron acceptor. Just a little quick review of ATP. It is a three-part molecule made up of adenine, which is the nitrogenous base, a 5-carbon sugar ribose, and then 3 phosphate groups bonded to the ribose sugar. It's bulky and does have a negative charge overall, and it tends to repel electrostatic charges, and this imposes a strain between the last 2 phosphate groups, causing high energy bonds to form. And the removal of these outermost phosphates then releases a huge amount of free energy that the cell can do. So usually these high energy bonds are represented by the little tilde sign, and this is simply because phosphate and oxygen tend to push each other apart, and these electrons are really moving in these bonds. And so when we separate the third phosphate off, we get a huge amount of energy released. If we should separate the second one here, we get not quite as much energy, but more energy than normally would be found in a normal covalent bond. ATP is the primary energy currency of the cell. In fact, it is the only form of energy that a cell can use to do work. When it is used in a chemical reaction, then it must be replaced. ATP utilization and replenishment is an ongoing cycle in all cells. We can create ATP from ADP and phosphate through two processes. Substrate level phosphorylation, which is the generation of ATP through the transfer of a phosphate group from a phosphorylated compound directly to ADP. Or we can do oxidative phosphorylation, and this is through the series of redox reactions that occur during the final phase of the respiratory pathway. Photophosphorylation is really a third form, and this is where ATP is formed through a series of sunlight-driven reaction in phototropes in the process of photosynthesis. So the loss of electrons is what? Oxidation or reduction, and the gain of electrons is which? True or false? When electrons are transferred in oxidation and reduction, a hydrogen ion will be transferred as well. And the most common electron carrier that we find in cells is what? The two ways that ATP is formed from ADP are what type of phosphorylation and which type of phosphorylation. So let's look at metabolism in energy production. And there are three basic catabolic pathways. Aerobic respiration, where the final electron acceptor will be molecular oxygen. Anaerobic respiration, where it will be something else besides besides molecular oxygen. And fermentation, where really the final products will either be an alcohol or an acid. Glycolysis is the most commonly used mechanism to break down carbohydrates, and the carbohydrate that we're primarily going to discuss is going to be glucose. So, kind of the overview of aerobic respiration. It has three primary stages: glycolysis, the citric acid cycle or Krebs cycle, and the electron transport with O2 as the final electron acceptor. Aerobic respiration also includes glycolysis, citric acid cycle, and electron transport, but the final electron acceptor is a non-oxygen compound such as sulfate, nitrate, or even bicarbonate. And then fermentation uses only the process of glycolysis. It does not contain the citric acid cycle or an electron transport. And the fermentation uses organic compounds as electron acceptors. And the two organic compounds that we see are alcohols and acids. Aerobic respiration, in using one molecule of glucose, will give us a potential yield of 38 ATP. Anaerobic respiration can give us anywhere from 2 to 36 ATP depending upon the electron transport system that we see in the anaerobic pathway. But for each glucose, fermentation only gives us 2 ATP because we are only using glycolysis in fermentation. So let's start with aerobic respiration. And this is a series of reactions that converts glucose to CO2 and water and allows the cell to recover significant amounts of energy that it will use to take ADP to ATP. Again, 3 primary steps, but I'm going to put in a 4th process here. So the first set of enzymatic reactions we're going to see in aerobic respiration will be glycolysis. Secondly, there will be an intermediate step, which this is kind of the odd 4th one. The citric acid cycle, also known as Krebs, and the electron transport chain, with molecular oxygen being the final electron acceptor. And we're going to go through each step so that you can see what's happening. Fermentation uses only glycolysis, and facultative and aerotolerant anaerobes will use fermentation uh, when no oxygen is present. So oxygen is not required for the fermentation process. It uses organic compounds, again, either an alcohol or an acid, or sometimes both, as electron acceptors. Anaerobic respiration, and those that can do it, will utilize glycolysis and the citric acid cycle and an anaerobic electron transport chain, where again molecular oxygen is not the final electron acceptor, but it will be something else, such as nitrite, sulfite, or even bicarbonate or other oxidized compounds, but not O2. So let's look closer at aerobic respiration and the steps. So, aerobic respiration, a series of enzymatic catalyzed reactions where electrons eventually are transferred from fuel molecules, food, to oxygen as the final electron acceptor, giving up energy along the way that is used to convert ADP to ATP. It is the principal energy yielding scheme for aerobic heterotrophs. It provides ATP and metabolic intermediates for other pathways. So let's start at the top, glycolysis. Glycolysis is where glucose, a 6-carbon structure, is enzymatically converted to a 3-carbon structure called pyruvic acid or pyruvate. For each glucose, we're going to get 2 pyruvic acids. Glycolysis occurs in the cytoplasm of all cells, and it is going to not require any oxygen. So glycolysis by itself is anaerobic in overall function. However, because it leads into the rest of the aerobic respiration, we need to include it here in the aerobic pathways. It does synthesize a small amount of ATP anaerobically, and pyruvic acid is an essential intermediate metabolite that will go on into the citric acid cycle. So here we have the picture. So the first step of glycolysis will be where glucose is actually converted enzymatically to glucose 6-phosphate. And you'll notice here that we actually use an ATP. There is an enzyme called hexokinase, hexa meaning 6, so the 6-carbon glucose, and it will actually take a phosphate from ATP, put it on the glucose to form the glucose 6-phosphate, and then we get out an ADP. Glucose 6-phosphate is then converted to its isomer called fructose 6-phosphate, and then in the third step we do a second ATP for adding another phosphate to the fructose, so we wind up with fructose 1,6-diphosphate. But we still have 6 carbons in our fructose molecule. It is at the fourth stage that we now split the 6-carbon structure into 2 3-carbon structures, one called glyceraldehyde 3-phosphate or G3P, the other one dihydroxyacetone phosphate, which just happens to be an isomer of G3P. So what we'll see is that this DHAP will be converted to a second molecule of glyceraldehyde 3-phosphate. This is what we call the energy investment stage, because we actually had to use 2 ATP. We then are going to take our 2 glyceraldehyde 3-phosphates and go into the energy production part of glycolysis. So each glyceraldehyde 3-phosphate will undergo a series of enzymatic reactions to go down to pyruvic acid. In that process, when we go from glyceraldehyde 3-phosphate to this molecule, we are going to reduce NAD plus to NADH. We're going to generate an ATP right here. We're going to get out some water, and we're going to generate out a second ATP. But remember, we're going to do this twice because we have two glyceraldehyde 3-phosphate. So we'll generate out another NADH right here, a third ATP, and a fourth ATP, and two pyruvic acid molecules. So we make a total of 4, but we use 2 in the energy investment stage, so our net gain of ATP from one glucose going through glycolysis is going to be 2. We're going to generate out 2 NADH molecules, and we're going to get a couple of water molecules out of this. Now, pyruvic acid can be utilized in several pathways by many different organisms. Strict aerobes and some anaerobes send pyruvic acid to the citric acid cycle for processing and further energy release, and that's what we're going to talk about. Facultative anaerobes will utilize pyruvic acid into acids or into other products. So, pyruvic acid can go to several different things. It can be a building block of some of the amino acids. It can actually build sugars, and it can build some of the fat metabolites. Or it can be converted through fermentation to acids along with some gas, or through fermentation to alcohols. But what we're going to look at is how pyruvic acid will be converted to acetyl-CoA and then enter into the citric acid cycle. So pyruvic acid must first be converted to acetyl coenzyme A. Pyruvic acid has 3 carbons, while acetyl-CoA has 2 carbons. And this will actually release the first of our carbon dioxide molecules. A cluster of enzymes and coenzyme A dehydrogenate pyruvic acid to the 2-carbon acetyl group. NAD plus will be reduced to NADH in this intermediate step before citric acid cycle. The NADH from both glycolysis and the from this intermediate step will then be shuttled to the electron transport that we'll talk about here in just a moment. All reactions that occur in the citric acid cycle happen twice in aerobic respiration because for each glucose molecule that started glycolysis, we're going to wind up with 2 pyruvic acids at the end and convert those both into 2 acetyl-CoA. The pyruvic acid will serve to transfer the energy stored in acetyl-CoA to NAD plus and FAD plus by reducing them to NADH and FADH2 in the citric acid cycle. So the main products of the Krebs cycle or citric acid cycle will be a production of 6 NADH for each original glucose and 2 FADH2 molecules. And we get 2 freebie ATP produced through substrate level phosphorylation. So here's the cycle. And the citric acid cycle occurs in the cytoplasm in prokaryotic cells and in the mitochondrial matrix in eukaryotic cells. So again, pyruvic acid in the intermediate step must first be converted to acetyl-CoA. We're going to generate out an NADH molecule for each pyruvic acid and a molecule of CO2. These 2 carbons that we generate from the CO2 molecules are 2 carbons that originated in our glucose. So the NADH molecules will go to electron transport, and the 2 CO2 molecules that are generated from the 2 pyruvic acids will actually be just waste products. And then we have 2 acetyl-CoAs. Acetyl-CoA will enter into the citric acid cycle and combine with a 4-carbon structure called oxaloacetic acid. Oxaloacetic acid and acetyl-CoA produce citric acid, hence the citric acid cycle. Citric acid then will be converted to a series of intermediate products that we go around the wheel, but malic acid, you'll notice, is converted back to oxaloacetic acid, hence the citric acid cycle. As we turn the wheel, from this reaction right here, we'll generate out another NADH and a CO2 molecule. In the next step, another NADH and CO2. At this step right here, we actually will generate out ATP. From this one, we get one FADH2. We put some water in here, and this actually makes this aerobic. And then when we go from malic acid to oxaloacetic acid, we generate out another NADH. So for each pyruvic acid, we get 1, 2, 3 NADHs, 1 FADH2, and 1 ATP. But because we run the wheel twice, we're going to get a total of 1, 2, 3, 4, 5, 6 NADHs, 2 FADH2s, and 2 ATP. Now, we still have energy stored in the electron carriers of NADH and FADH2. These will now go to the electron transport chain. And this is a chain of specialized redox carriers that receives the electrons from NADH and FADH2, transferring the electrons along the chain and creating a hydrogen ion gradient across a cell membrane. The electron transport chain in prokaryotic cells occurs at the cell membrane, or I should say across the cell membrane. In eukaryotic cells, electron transport occurs across the inner mitochondrial matrix or across the cristae. These electrons are passed sequentially from one redox molecule to the next, and this allows a flow of electrons and hydrogen ions across the membrane, creating this hydrogen ion gradient. Oxygen will be the final receiver of the hydrogens and electrons, and the oxygen, the hydrogens, and the electrons will form water. So here's what it looks like. Now, this is a busy slide, so let me just go through it. We have a series of these carriers, and FMN is actually related to FAD, but this is the first carrier in most cells. Then we have ubiquinones. Ours is actually known as coenzyme Q. And then we have a series of proteins called the cytochromes. At the end of the electron transport chain in the membrane, either, again, the cell membrane in prokaryotes or the cristae membrane in the mitochondria of eukaryotes, sits this enzyme called ATP synthase. So here's what happens. NADH, along with its hydrogen ion, will come in and give up its electrons and hydrogens to FMN. The electrons will actually then be shunted to coenzyme Q, and the hydrogens will be shunted across the membrane. The electrons then will move down from coenzyme Q to cytochrome B, and don't worry right now about this, but we have hydrogens over here that will also be then shunted across as these electrons from the NADH are moving along. It goes through cytochrome C to cytochrome A to A3, and then again we pump a third pair of hydrogens across the membrane. What we now have is a higher increase of hydrogen ions over here, less over here. The ATP synthase is going to do two things. One, it will be a transport channel for these hydrogens. The second thing is, as these hydrogens move through this enzyme, it generates a rotational power is the best way to describe it. And what that does is create or transfer the energy of the movement of the hydrogen ions to form the bond between a phosphate and ADP to generate out ATP. The hydrogens and the electrons that exit the chain will then combine with the oxygen to form our water as the final electron acceptor. Each NADH pumps 3 sets of hydrogens across, and that means that each pair will give us an ATP. So for each NADH, we get 1, 2, 3 pairs. That means we get 3 ATP. Now, if you go back, we got 2 NADHs from glycolysis. We got 2 NADPHs, or sorry, 2 NADHs from the intermediate step in the conversion of pyruvic acid to acetyl-CoA. And then in the citric acid cycle, we got 6. So what that means, if every NADH that was created from the one molecule of glucose gives up its electrons and hydrogens to this chain, we will actually generate out 30 ATP for all of the NADPH that was produced. Now, FADH2, however, skips the first electron carrier. It gives up its electrons and hydrogens to coenzyme Q, and then this actually will be shunted, and so we get 1 pair there and 1 pair there. So for each FADH2, we only get this pair and this pair of hydrogens. That means for each FADH2, we get 2 ATP. We produced 2 FADHs in the citric acid cycle, so that means from FADH2 we get a potential of 4 ATP. That means altogether that we have a potential of 30 from NADH and 4 from FADH2. That means we have a potential of 34 ATP from aerobic electron transport. ATP synthase will be stationed along these membranes in close association with these electron carriers, and it captures that released energy of the hydrogen ions from the electron transport carriers. This is what's then known as oxidative phosphorylation. It is the coupling of ATP synthase to the electron transport. And again, for each NADH that gives up the electrons and hydrogens to the chain, we will give rise to 3 ATP molecules through ATP synthase. Now, since FADH2 and NADH enter the electron transport at different points, there's less energy generated by the FADH2s, and therefore, again, each FADH only gives rise to 2 ATP molecules. So this whole process of the electron transport is a process actually known as chemoosmosis. What this simply means is that as the electron transport carriers shuttle the electrons along the chain, the hydrogen ions are being actively pumped either into the periplasmic space of the mitochondria or the space between the cell wall and the cytoplasmic membrane of the prokaryotic cells. This sets up that concentration gradient of hydrogen ions, and this is known as the proton motive force. And it is the proton motive force then that consists of a difference in the charge between the outside of the membrane, which will be positive, and the inside, which will be negative. The hydrogens would like to go across these membranes, but can't because of their charges, can't get through the phospholipid membrane. So they have to go through ATP synthase. This sets the stage then for the ATP synthesis. Now, ATP synthesis in eukaryotes occurs through the same overall process. The electron transport chain is stationed in the inner mitochondrial membrane between the mitochondrial matrix and the outer inner membrane space. This difference affects the amount of ATP that can be produced. But again, here's what I want you to understand about this. In aerobic respiration, the first stage, glycolysis, does not require oxygen. but we have to get the product of glycolysis, pyruvic acid, in order to do the rest of aerobic respiration. For each glucose, through glycolysis, we get 2 NADHs and a net gain of 2 ATP. So we get a yield of 2 ATP. Remember, we produced 4, but we used 2, so we're going to have to pay back the 2 we used. And we get the 2 pyruvic acids. But we also produce 2 waters. Pyruvic acid converted to acetyl-CoA gives us 2 more NADHs and 2 CO2s. Acetyl-CoA enters the citric acid cycle. For each glucose, original glucose that we start with, citric acid cycle gives us 6 NADHs, 2 FADH2, 2 ATP, and a total of 4 CO2 for each glucose. So we have 4 CO2 here, 2 there, 4 plus 6 carbons equals the 6 carbons of glucose. We also will see 2 waters that are consumed in the citric acid cycle. So basically, we wipe out the waters that we produced here in glycolysis. Then the 1, 2, 3, 4, 10 from NADHs will go to the electron transport chain, and the 2 FADHs will go to the electron transport chain. If everybody gave up the electrons and hydrogens to the electron transport chain, we would then get a total of 34 ATP from the ETC. We also will see that we will produce 6 molecules, we use 6 molecules of oxygen, and we produce 6 molecules of water. So our potential yield from one glucose that starts up here is 38 ATP. Now, will all of the NADHs and FADH2s give up electrons to the electron transport chain? Probably not. And so what we'll find is that we actually will get somewhere usually total, somewhere between 28 to 32 ATP produced. But we could actually write this equation as 1 glucose plus the 6 oxygens goes to 6 CO2s and 6 waters. And then our ATP is produced. So the total possible production from 1 glucose in aerobic respiration is 40 total. 4 from glycolysis, 2 from the Krebs cycle or citric acid cycle, 34 from electron transport, but again, we have to pay back the 2 that were expended in the energy expenditure stage of glycolysis, and so therefore we can get a maximum optimal total of 38 ATP per 1 molecule of glucose. Now, if you know of somewhere where I can put in $1 and I'm guaranteed to get $38 back out, I would like to talk to you as soon as possible. But again, the actual total ATPs produced are probably lower, and especially eukaryotic cells, because there is some energy that is expended transporting the NADH and FADH2s across the mitochondrial membrane, as well as we may use some of the intermediate products for other things. So we have 6 CO2 molecules that are generated during the intermediate step in citric acid cycle, and these are simply waste products. We will consume 6 oxygen molecules during the electron transport, and will produce a total of 6 water molecules altogether. The terminal step of the electron transport chain is actually catalyzed by an enzyme called AA3, also known as cytochrome oxidase. This is adapted to receive the electrons from cytochrome C, pick up the hydrogens from the solution, and react with the molecular oxygen to form the water. Now, anaerobic respiration uses still glycolysis, citric acid cycle, and an anaerobic form of electron transport. Nitrate and nitrite reduction systems are found in several bacteria, but the most common one that we see is E. coli. A nitrate reductase catalyzes the removal of oxygen from nitrate, NO3, reducing it to nitrite, NO2, and water. So the final electron acceptor is actually nitrate. We again also can see sulfate and bicarbonate systems. And a test for the nitrate to nitrite is one of the physiological tests we actually use in identifying bacteria. Denitrification, these enzymes can further reduce nitrite, NO2, to nitric acid, NO, nitrous oxide, N2O, and even nitrogen gas. And these are found primarily in the Pseudomonas and Bacillus genus of bacteria. This is an important step in recycling nitrogen back into the biosphere environment. Now, fermentation is anaerobic. It's the incomplete oxidation of glucose or other carbohydrates in the absence of oxygen. It only uses glycolysis, and it uses organic compounds as the terminal electron acceptors. It yields a small amount of ATP, in fact, actually 2 for each glucose used. So we've already gone through glucose, and we know that we've gone through glycolysis, and we know that glucose is converted to 2 pyruvic acids. And with that, we get 2 NADH. But what happens is that when all of the NAD pluses get filled up, glycolysis stops. And so we don't even produce the 2 ATP that we get out of glycolysis. So what fermentation does is kind of unique. It will take pyruvic acid and the NADHs and convert them to something else and oxidize NADH back to NAD+. So pyruvic acid could go to something like lactic acid, which will then free up the NAD+, and now we can go back up here and start with another glucose, go through glycolysis, and generate out our 2 ATP. Or pyruvic acid could be converted to acetaldehyde and eventually to ethyl alcohol or another alcohol using, again, NADH, where it is oxidized back to NAD+. So this just keeps glycolysis going and going and going and going. But again, we're only going to get 2 ATP for each glucose that is actually used. So all fermentation does is free up the NAD plus to keep glycolysis running. And it produces acids and or alcohols depending on what enzymatic systems the bacteria have. We actually have the enzymes that will take pyruvic acid to lactic acid in our muscle tissue. And this is what causes the burn in those athletes that run out of energy. Alcoholic fermentation occurs in yeasts and some bacterial species that can have metabolic pathways for converting that pyruvic acid to ethanol. Decarboxylation of the pyruvic acid to acetaldehyde then will be converted to the ethanol. NADH formed during glycolysis is oxidized to generate out NAD+, and again this allows glycolysis to continue as long as there's glucose present to be used. Acidic fermentation, these pathways vary extremely depending upon the species. Homolactic fermentation only lactic acid is going to be produced from the pyruvic acid. This is what our cells can do when there's no more oxygen present to run citric acid cycle and electron transport. But other organisms also can do heterolactic fermentation, where glucose can be fermented to a mixture of lactic acid, acetic acid, and even some other acids along with carbon dioxide. Mixed acid fermentation can be done by members of the family Enterobacteriaceae, which includes a broad number of the gram-negative bacteria. They possess enzyme systems that convert pyruvic acid to several acids simultaneously. So acetic acid, lactic acid, succinic acid, formic acid, along with CO2 are produced. This fermentation activity accounts for the accumulation of some types of gases that we see actually in the intestines, because E. coli is one of the Enterobacteriaceae. Some bacteria reduce organic acids to neutral 2,3-butadiene-diol and other solvents. And in fact, this butadiene-diol, it's hard to say, is actually a form of alcohol. So we can actually see both acids and alcohols produced at the same time. Now, can we use lipids as an energy source? And certainly we can do that. We can break apart the fatty acids joined to the glycerol. The glycerol is a 3-carbon sugar that can be converted to that dihydroxyacetone phosphate, which will then be converted to G3P. Beta oxidation of the fatty acid chains actually slices off 2 carbons at a time, and those 2 carbons will actually be converted to coenzyme A, creating acetyl-CoA that can enter the citric acid cycle. Oxidation of a 6-carbon fatty acid then would yield 50 ATP compared to 38 for a 6-carbon sugar. However, even though we get plenty of ATP, there is a lot of front-end processing, and so we actually wind up with less energy overall in using fats than carbohydrates. That's why carbohydrates are a primary energy source, while lipids are a secondary energy source. We could use a protein for energy. We don't like to do this. We'd rather use the protein for building blocks. But proteases will break down proteins to their amino acid components. And in the amino acid pool, we can actually pull some of those amino acids and remove the amine group through deamination. This deamination must take place in order to use amino acids for energy production. The remaining carbon compound that can be easily converted to one of several of the citric acid intermediates. However, again, we're going to lose a lot of energy production by using proteins as energy. So what we want to do is not use proteins for energy if we can help it. So for example, glutamic acid, one of our amino acids, we have to take that amine group off. And we'll do that by converting it to a carbohydrate called alpha ketogluteric acid and ammonium ion. NH4 plus will be converted to NH3, which is ammonia. So let's kind of put it all together. So carbohydrates, primary energy source. We'll go through glycolysis to pyruvic acid. Pyruvic acid can be converted to acetyl-CoA if oxygen is present, and we have the enzymes to do it. We'll go into the citric acid cycle and electron transport. Fats can be broken down into the fatty acids, and this chart doesn't show it, but we can actually convert again that glycerol backbone to pyruvic acid through the intermediate of glyceraldehyde 3-phosphate. The fatty acids will be converted to acetyl-CoA. Does the citric acid cycle care where the acetyl-CoA comes from? The answer is absolutely not. And so this will run the citric acid cycle and electron transport, but again, we may not get as much produced out of the fats. And from their nitrogen pool of our amino acids, some of the amino acids can be converted to acetyl-CoA, some can be converted to pyruvic acid, some will be converted to some of the intermediates within the citric acid cycle. Now, the nice thing that I like about this summary is that it also shows that we can use some of these intermediates for other things within the cell. So, when we take glucose to glucose 6-phosphate, we could undergo glycolysis for energy, or we could store that glucose 6-phosphate through the process of glycogenesis as glycogen in muscle cells or in our liver cells. When we then need more ATP, we can break the glycogen down, which is nothing more than just a string of glucoses strung together, through the process of glycogenolysis, and then go through glycolysis. Pyruvic acid actually can be converted back to glucose through gluconeogenesis. Fats or acetyl-CoA can actually be converted back to fats through the process of lipogenesis. So let's say I eat my Snickers bar today. Am I going to need all of that ATP from all of the glucose? Well, probably not. So once we get down to the acetyl-CoA, if I don't need a whole bunch of ATP from citric acid and electron transport, I have enzymes that take that acetyl-CoA and convert it to fat where it's stored in the adipocytes. And I evidently have really good enzymes that do this lipogenesis. Now, you'll notice then that some of these are going to be recyclable. We can go the opposite direction. Now, I will mention this right over here, the urea cycle. In a few bacteria, they can actually take that ammonia that was produced from the deamination process and undergo what's known as the urea cycle. Along with CO2, this will actually then produce some energy along with the waste product of urea. And some of the intermediate products can actually then go into the citric acid cycle. And some of those can actually go into the urea cycle. This urea cycle only occurs in a very few bacteria that have the enzymes to actually do it. So, true or false, glycolysis requires oxygen. True or false, the citric acid cycle requires oxygen. True or false, the electron transport chain requires oxygen in anaerobic respiration. Or is that in aerobic respiration? And true or false, fermentative organisms that only do fermentation, grow more slowly than aerobic organisms. And cells can potentially produce how many molecules of ATP per molecule of glucose used in aerobic respiration? Now, the cell can actually be very frugal in energy production, and this is known as amphibolism. Most catabolic pathways contain strategies or strategic molecular intermediates that can be diverted into anabolic pathways, such as the glucose 6-phosphate to make glycogen. A given molecule then can serve multiple purposes to derive maximum benefit from all nutrients and the metabolites throughout all of these enzymatic reactions. Catabolic and anabolic pathways are integrated to improve efficiency of building the blocks that we need for structures and other things, as well as production of energy. And again, this just kind of shows the same thing. So, could I take intermediate products of glycolysis and take them back to carbohydrates, where I could then use the carbohydrates to build the macromolecules of starch and cellulose? And this can be for cell walls in the eukaryotic cells or storage. Starch is a storage form of carbohydrates. Can I take acetyl-CoA and make fats? And absolutely, and this can be a storage form of energy when I no longer have the carbohydrates. Can I actually take those amino acids instead of using them in the pyruvic acid or to make pyruvic acid or the intermediates here of the citric acid cycle? Can I use the amino acids from my pool to make proteins for enzymes, membranes, and other structures? And I can use those amino acids also to make nucleotides that I'm going to need to make my nucleic acids, DNA and RNA, to build chromosomes and do the genetics of the cell. So a precursor molecule is a compound that is a source of another compound. For example, G3P, glyceraldehyde 3-phosphate, can be diverted from glycolysis and converted into precursors for amino acids, carbohydrates, and even fats. Pyruvic acid is a precursor in that it provides intermediates for some of the amino acids that we can actually make. It can provide gluconeogenesis for glucose synthesis in the event of inadequate glucose supply. Acetyl-CoA can be a precursor for several amino acids as well as lipids. Precursors to DNA and RNA, pathways that synthesize purines and pyrimidines, originate actually in the amino acids. And this can be dependent upon intermediates from the citric acid cycle. During times of carbohydrate deprivation, carbohydrate starvation, organisms can convert amino acids to intermediate of the citric acid cycle and derive energy from proteins. That's why diets like the keto diet and the Atkins diet work for short periods of time. The problem is, again, when we use amino acids to produce energy, we have that deamination process that must happen. That produces huge amounts of ammonia, and that can actually be very hard on the kidneys because the kidneys have to take that ammonia out of the body. Carbohydrate biosynthesis, really the crucial role of glucose in metabolism and energy utilization, is that we want to make sure that we have plenty of carbohydrates on hand when we need them. So biosynthesis can be ensured by several alternative pathways. The major component of cellulose cell walls in plants and starch storage granules are the simple carbohydrates. So glucose 6-phosphate can be used to form glycogen. The proteins are a large proportion of the cell's constituents. Remember, when we looked at the dry weight of an E. coli cell, 50% of the dry weight was actually protein. Proteins are essential components of the enzymes, cell membranes, cell wall, and cell appendages, if we have any. In eukaryotic cells, 20 amino acids are needed to make all of these proteins, and we're going to talk about protein synthesis a little later on. Some organisms, such as E. coli, have pathways that can synthesize all 20 amino acids within the cell from other precursors. In our cells, we can only actually make 11 of these 20 amino acids. The other 9 are what we call essential amino acids because we must get them from the diet. Now, cell division. Well, in order for the cell to divide, not only do we have to copy the genetic code, but we need twice as many ribosomes, mitochondria, enzymes, etc. The cell wall and cell membrane has to nearly double in size. The phospholipid bilayer components assemble spontaneously and require actually no energy input. But proteins and other components must be made with energy input before they're added to the membranes. Cell wall growth requires energy input since the cell walls are made up of carbohydrates or proteins. So which of the following does not serve as a precursor molecule? True or false? Amino acids can be converted into intermediates of the citric acid cycle. And true or false? E. coli can synthesize all 20 essential amino acids. All right, let's talk about the last thing here, and that is photosynthesis. Photosynthesis can be divided into two main groups of reactions, the light-dependent reactions and the light-independent reactions. The light-dependent reactions proceed only in the presence of sunlight. They are catabolic, energy-producing reactions. They produce an electron carrier called NADPH and ATP. The light-independent reactions, also known as the Calvin cycle, are going to take the NADPH and ATP made by the light-dependent reactions and actually use those to make glucose. They are anabolic, therefore, and synthetic reactions. Carbon atoms from CO2 are added to the carbon backbones of organic molecules in order to make glucose. So here's what happens in photosynthesis. In photosynthetic organisms, we have a protein called chlorophyll. Most photosynthetic organisms have two photosystems, photosystem 2 and photosystem 1. Both of these are going to be energized by sunlight, and that excites electrons within the chlorophyll. And what we actually see is an electron transport chain. Now, in eukaryotic cells, the chlorophyll is found in the thylakoids within the chloroplast. And so this is what's going to happen across the membrane of the thylakoid. Or if we're talking a prokaryotic cell, these reactions will actually occur across the cell membrane. What we're going to see is as the sunlight excites electrons of the chlorophyll, those electrons will then be passed down a series of electron carriers, which will create ATP and NADPH. The light-dependent reactions occurs in the thylakoids, and they use that to capture energy from sunlight and create chemical energy in the form of ATP and NADPH. Water travels into the cell from the system, from the roots in the plant or from the environment, and that water then will be used to convert the oxygen that comes in to ATP. Then we take the ATP and NADPH from the light-dependent reactions, and then we can put it into the light-independent reactions, and this is known as the Calvin cycle. This occurs in the stroma of the chloroplast or in the cytoplasm of the prokaryotic cell. CO2 will enter the cell or the chloroplast, where we actually will add that CO2 to a 5-carbon structure. And as we go around the Calvin cycle, G3P will be produced. We can take 2 of those G3Ps and make glucose. So here is the chemical reaction. Light energy plus water plus CO2 will go to oxygen and carbohydrates. This is known as photophosphorylation in the light-dependent reactions, and it's very similar to the electron transport that we saw in the eukaryotic cells. Channels in the thylakoid pump hydrogen ions into the inner chamber of the thylakoid, producing again a hydrogen ion gradient across the membrane. ATP synthase is sitting at the end of the chain, and this will phosphorylate ADP to ATP as the hydrogens move through ATP synthase. NADPH or NADP will be reduced to NADPH, and NADPH and ATP are released into the stroma of the chloroplast, where they then can drive the reactions of the Calvin cycle. So when light activates photosystem 2, and it is the first photosystem that is actually activated, it sets up a chain reaction in which electrons are released from chlorophyll. Those electrons then are transported along a series of carriers through redox reaction to photosystem 1. Once we reach photosystem 1, the empty position in photosystem 2 is replenished by photolysis of water. So we're going to use water, and what we're going to get is hydrogen ions and O2. The O2 will be a waste product. Then, as we go through the chain, we're going to pump hydrogen into the interior of the thylakoid, and this produces the condition in which ATP synthase can then run. The final electron and hydrogen acceptor is NADP, which it receives these from photosystem 1. So both NADPH and ATP are released into the stroma of the chloroplast where they can feed into the Calvin cycle. So photons of light energy hit photosystem 1. It breaks up water, pumps the hydrogen across the membrane, creating that osmotic force. We then will carry the electron to photosystem 1, where the electrons then will be transferred to NADP and reduces NADP to NADPH. As the electrons move through the ATP synthase, we're going to then make ATP. The light-independent reactions, and these used to be called the dark reactions, and that's really a misnomer. These occur whether light is there or not. But this occurs in the chloroplast stroma or in the cytoplasm of the bacteria. It uses energy, the ATP and NADPH, produced in the light reactions to synthesize glucose, which can then take the glucose and go through aerobic respiration to make more ATP. So, this is what will happen. In the Calvin cycle, and just for the moment accept that we have this molecule called ribulose 1,5 biphosphate 5-carb. It's a 5-carbon structure. We call it ribisco. We're going to take the carbon from CO2, add it to the 5-carbon structure so that we have a 6-carbon structure. We're going to split that into two 3-carbon structures. We're going to use ATP to put some phosphates on those. And eventually what we're going to do is then use hydrogens from NADPH and oxidize it to NAD+, and we're going to produce glyceraldehyde 3-phosphate. Two of those can then be made into glucose through enzyme systems. But we can also use the G3P to generate back out the ribisco using ATP. So we have a series then here of 7-carbon and 5-carbon intermediates, and we're going to talk about those. So the Calvin or light-independent reactions uses the ATP, NADPH we produced in the light-dependent reactions using CO2 to produce glucose. So when the plant's in the dark, can it use glucose then to make its ATP through aerobic respiration? And the answer is yes. Now, if photosynthesis releases oxygen from the breakdown of water, it is known as oxygenic photosynthesis, and this is the dominant type of photosynthesis that occurs on Earth. This occurs in plants, algae, and the cyanobacteria. However, some photosynthetic users actually are anoxygenic. And this occurs in the green and purple bacteria that use a specific chlorophyll known as bacteriochlorophyll. They only have the cyclic photosystem 1. They create a smaller amount of ATP, and they use hydrogen gas or hydrogen sulfide or elemental sulfur as electron sources instead of water. So no oxygen will be released. These are mostly strictly anaerobic organisms. So last concept check for this chapter. ATP is produced through the light-Watt reactions of photosynthesis. Glucose and other molecules are synthesized using the light-Watt reactions of photosynthesis. The green and purple bacteria utilize what as a light-capturing pigment? And finally, archaea utilize what as light-capturing pigment? And you may have to do some digging on this last question. So, there's Chapter 8. See you next time.